And he's going to talk today about live attenuated pertussis vaccine to prevent disease, colonization, and transmission. And this is the person who developed this, Camille Lott, has been a friend of mine for over 20 years. And so, Keith, you have to do him justice.
I'll do my best. So ILiAD Biotechnologies, we're a clinical stage company. The vaccine we're developing is BPZE1. It's in phase 2. And our general approach has been to address infectious diseases that are insidious, quiet. People may not know they have them, and yet they can still cause pathology. So hepatitis C would be an example of that. HPV would be an example of that. And we believe also pertussis has that characteristic. In the case of pertussis, it's also underappreciated. Most of the cases are not reported, particularly on the subclinical side. And so Bordetella pertussis is our first target. Why pertussis? First of all, the current vaccines target the disease. They do not target the infection. Infection being nasopharyngeal colonization. And it is basically axiomatic in public health that if you really want to slay the dragon, solve the problem truly, once and for all, you have to get at transmission. And even better if you can get at the reservoir. The current vaccines, and even the old host cell vaccine, did neither. And we will show you evidence and continue to generate that evidence that BPZE1 has the capability to do that. Whooping cough is currently the most poorly controlled vaccine-preventable disease of children. So even though in the United States we have 90+% vaccination rates, it's still poorly controlled. And we see this globally in many countries. Despite that the vaccines that have a pertussis component are suboptimal, they still generate a lot of revenue, more than $5 billion a year. And this organism, Bordetella pertussis, secretes a number of toxins. And these toxins are highly biologically active. They're used in many preclinical experiments for other diseases. And so we're paying attention to that, and that's important to us and important to our sense of what this vaccine ultimately is going to be able to contribute. We identified this vaccine in 2012, and we licensed it from the Institut Pasteur in 2013, again developed in the lab of Camille Lott with Natalie Milkarek and their team, and they remain strong collaborative partners, and we do a lot of science together, and we're very grateful for that relationship. In 2012, 13, and 14, we had the highest rates of pertussis in 50 years in the United States, even though we vaccinate more than 90% of the population. So this is a problem. This is really fundamentally not what you want from a great vaccine. And the CDC has said, and we've heard it from other public health agencies, but this is a quote from them, as long as currently available acellular pertussis vaccines are in use, it's likely the new normal will be higher disease incidence throughout pertussis cycles. The translation is, we need a new pertussis vaccine. Okay, a little bit about the organism. It's a gram-negative organism. It's highly infectious, very high attack rates. It's transmitted by cough or sneeze or just talking, or even just walking into a room where a person with whooping cough is lying in a bed. You can pick it up just from the air. It transiently colonizes the respiratory tract, and it causes roughly 20 million symptomatic infections a year, 200,000 deaths. Again, the data depends on where you look at it, but it has a major, horrific public health impact globally. And if you're an infant and you get this infection, you have a 50% chance of being hospitalized. So it's a pretty horrific disease and a clear problem globally and in the United States as well. Now, that is all well known. What is underappreciated is that subclinical colonizing infections, infections that basically colonize the nose and nasal passages, but never get reported, they never get diagnosed, there's not much consideration about what pathology, if any, that may contribute to. those are enormous, in the hundreds of millions. We don't know for sure. But if you look at the data cross-culturally, you see this over and over again, and we'll talk about it in just a moment. So with respect to these infections, most people know about whooping cough, but most people don't know about some of the potential consequences of a whooping cough infection. So this was a study that was published by Berger and et al. In November of 2017, last year. And it was a robust study, 25 clinical sites funded by the NIH. And here's what they found. If you were in the pediatric ICU with a pertussis infection diagnosed, and you left the pediatric ICU, there was not a difference between the controls and the patients who had pertussis. However, 1 year later, more than a third, 37% of 1-year-olds who had a pediatric ICU pertussis infection had a neurocognitive deficit. More than 1 out of 3. 9% had a neurocognitive deficit that was so bad it was 2 standard deviations from the mean. That is significant intellectual disability. So these are very, very serious infections. And remember what we said a moment ago, if you're an infant and you get pertussis, you have a 50% chance of landing in the hospital. So that is striking. But there was something that was more striking. And this was, I would say, not expected by the general community, but it's exactly what we expected. What they found was that there wasn't a symptom or a constellation of symptoms that you could determine that would indicate who was going to have that neurocognitive deficit a year later. You could have a high white blood cell count, you could have a low white blood cell count. You could have a fever, you could not have a fever, you could have a cough, you could not have a cough. None of them were able to determine whether you were going to have a neurocognitive deficit. And that was consistent with our point of view, which is that pertussis is a toxin-mediated disease, and it's these toxins that cause pathology. Now, there are other potential pathologies associated, but at the heart of it, we believe that these toxins are driving this kind of pathology. So it begs the question, if you have so many people walking around with a subclinical pertussis infection, infections that are still secreting these toxins, what are the implications of that? And it's an open question right now, and we're working on it. And I would say that to anyone who finds that question compelling, I would encourage you to work on it as well. There's a lot of work to be done. But we believe that there's a significant public health impact with respect to that. So, we wanted to make sure that whatever vaccine we were going to develop prevented subclinical nasopharyngeal colonizing infections. So are there subclinical infections? Yes. All over the world, we see it globally. The first study is in China. They took a single swab of pediatric patients in a non-epidemic period, and what they saw was 4.8% of those children had pertussis living in their nose and nasal passages. This was in a province in China that had 99% vaccination rates, acellular vaccine. So that's a single swab. In the US, we can talk about different studies, but in the study that Dr. Cherry was an author on board, in the control group, there were 10 million estimated cases on the basis of that control group at a time when the CDC was reporting 7,000 cases. In the Netherlands, 6.6%. In Italy, 14.1%. Dr. Cherry and I were talking earlier today. He had done another study showing 6% over 5 years. So it is pervasive. Subclinical pertussis infections are pervasive. And they're not reported, and they're not diagnosed, they're not treated, and most people don't think that there's much to them except for transmission. That's not our point of view. Okay, so from a practical whooping cough point of view, what's the impact of that? And this was a study that was done by the FDA, Werfel, Zimmerman, and Merkel. They created the baboon model of pertussis. And in that model, they did the following. They vaccinated a group of baboons with the acellular vaccine. Then they challenged them with virulent pertussis in their nose and nasal passages. And then they wanted to see what happened. and those baboons did not get whooping cough. And we know that. The acellular vaccine in the short term can do a decent job, certainly for baboons that have never been exposed. However, when they looked for colonization rates in those baboons, there were a huge number of organisms. And in fact, the baboons that got vaccinated had more organisms for a longer period of time than the baboons that didn't even get vaccinated. So that was important. So that was one point they made. The other point of this study was just as important. If you took a baboon that did not have whooping cough, just being a baboon, and you put it in the cage with a naive baboon, what you saw was that you could get transmission from the non-coughing, just regular baboon that happened to have the organisms in its nose and nasal passages. You saw transmission in the same cage. Then they did a different study. They put two separate cages with the colonized baboon in one cage and a naive baboon in another cage across the room with some laminar airflow, and once again they saw transmission. And so they came to the conclusion, which was frankly our point of view, and we loved seeing the confirmation of it, that this vaccine, the fact that acellular vaccinations do not block either colonization or transmission is the reason that we've had these tremendous increases in pertussis over the last couple decades. As I said in that slide that was kind of messed up, 2012, 13, and 14, the highest rates in 50 years in this country, even though we vaccinate more than 90%. That is not a great vaccine. Okay, so BPZE1. Why did we go here? We looked at several hundred technologies to try to solve the problem as we saw it. And we identified this beautiful vaccine that was being developed at the Institut Pasteur by Camille and his team. And it's a live attenuated intranasal vaccine, and it does two important things. It delivers systemic immunity, but as important, if not more important, it delivers excellent immunity above the neck, in the nose and nasal passages, unlike the acellular vaccine and even the whole cell vaccine. Now, it's genetically modified. The idea behind the vaccine was if you get a real whooping cough infection, you get over it. You get great immunity above the neck, you get great immunity below the neck, systemic and mucosal immunity. But of course you can't administer virulent pertussis to people because you're going to hurt people. So, what do? They knew the full genetic sequence of the organism, and they knew where these key toxins were on the organism, and they were able to modify them, either remove them or enzymatically inactivate them or change them. 15 years in development, we licensed it from the Institut Pasteur in 2013, and we now, as I mentioned, have a phase 2A study in progress. These three key toxins were the toxins that they were able to modify. Pertussis toxin, dermatonecrotic toxin, and tracheal cytotoxin. And essentially, BPZE1 is a living organism, and the pertussis toxin has been mutated in two places so that it's enzymatically inactivated. Dermatonecrotic toxin has been removed completely from the genome, and tracheal cytotoxin was reduced by changing out an AMP gene by technology I don't really understand, to be honest. But they were able to reduce the secretion of that by more than 98%. So how does it work? It's given intranasally. It colonizes. BPZE1 is given intranasally. It colonizes. It's a live attenuated bacteria. And then it clears. And in that clearance, the host has the induction of an immune response, both local mucosal and systemic. So that in the third image there, when you are challenged then with virulent pertussis, you're protected. That's the idea of the vaccine. Now, up until a few months ago, the literature was not clear on, well, how does it induce that type of mucosal immunity? And this paper was published. This is a paper that Louise Salons and Camille Locht and Kingston Mills and others on the team had published in the fall, this fall, just a couple months ago. And it was published in Nature Mucosal Immunology. And it's a very, very elegant series of experiments. Essentially, what they were able to determine was when you get a natural infection, or when you get a BPZE1 vaccination, you develop tissue-resident memory cells that start in circulation but make their way to the mucosa. These then trigger an IL-17 response, which trigger secretory IgA, which is protective. And what you see is that the IgA actually decays fast, But when those animals are then rechallenged, if they previously had a whooping cough infection or they've been given a BPZE1 vaccine, those tissue-resident memory cells proliferate. They secrete IL-17, which subsequently allow the induction and a robust production of secretory IgA, which is if you use knockout mice for IL-17 or knockout mice for SIgA, they're not protected. So this is a beautiful set of experiments. You do not see the induction of tissue-resident memory cells with acellular vaccines. So this is a beautiful paper I highly recommend for anybody who's interested to take a look at that. Okay, there we go. Okay, so remember the study that we talked about that the FDA did with the baboon model, and they vaccinated the animals and they challenged them? Well, when we in-licensed the vaccine, the very first study that we did was to do that same experiment. We went back to the same Oklahoma Primate Center, we went to the same principal investigator, essentially the same protocol, except they had used 3 doses of acellular. We used a single intranasal dose of BPZE1. And on the y-axis is essentially the total pertussis burden. That is how many bugs are in the nose until they're cleared. And on the that's the y-axis. And on the x-axis, you see that number for the acellular vaccine in blue, the whole cell vaccine in gray, and that small line there, BPZE1 in red. And what we were able to determine is that the reduction in nasopharyngeal colonization by BPZE1 compared to acellular was 99.8% less. 99.5% less than host cell, which was better than acellular. This wasn't a subtle difference. This was night and day. And the challenge in the BPZE1 experiment was 15 billion pertussis organisms. So the plane took off with 10,000 people when it only had to take off with 300 people. This is really a very, very powerful slide. And we, of course, were very excited, and it gave us a lot of confidence. In terms of our strategy, we're going to move to adolescents and adults first, both to protect them from whooping cough and also to protect them from getting colonized and transmitting the infection. And then we will move to neonates. There's much more work to do in the neonates for safety issues that are obvious. But in preclinical studies, the vaccine works beautifully. Given to neonates, it protects against challenge very, very well. In terms of just the adult and adolescent market, we've done mathematical modeling with Sam Scarpino and Ben Althaus. These are PhDs in mathematical modeling, and this paper is submitted for review now. But what we were able to see that if you use less than the current vaccination rates with BPZE1 as opposed to acellular, in 10 years you can reduce infant pertussis by 80%. That's the data. Now, in terms of our clinical data, we've had 2 phase 1 clinical trials that were completed at Karolinska in Sweden. Very clean safety profile, and as I'll show you in a moment, we're very excited about the immunogenicity. There were no vaccine-related serious adverse events, and in the highest dose group, we had a 100% immunologic response. So on the y-axis there is the percentage of seropositive subjects, and in the BPZE1 on the x-axis, it's showing the placebo group and different doses of BPZE1, 10 to the 7th, 8th, and 9th. And in the 10 to the 9th group, we had 100% seropositive responses. And of course, we were quite excited, and that then led to an application with Vanderbilt through the VTEU to the NIH, and now the NIH is funding our phase 2A study at Vanderbilt, first working with Kathy Edwards and subsequently with Buddy Creech. They've been terrific partners. And in addition to funding the clinical site, the NIH provides all the study management and monitoring. Now, in terms of the study design for phase 2A, it's 50 patients, 15 placebo, and then 15 in the 10 to the 7th and 10 to the 9th group. We're using a lyophilized product, which is a more commercially scalable product, as opposed to the phase 1s where we're using a liquid. And in the study, we're also using an atomizer called the Vaccinator made by Teleflex with a very strong portfolio at the FDA. And that will help increase mucosal coverage. We expect actually that will even improve our immunogenicity. We're also having 5 controls using just the syringe to make sure that the vaccinator is not affecting any profiles. In the animal studies, it worked beautifully. So we expect the same here. Once we have a successful phase 2A 1-month set of data, then we will move to our phase 2B, which will be a 300-patient multi-center study. Okay, so what do we have in conclusion? So the first thing is that Bordetella pertussis is a major public health problem. Both the well-known problem of whooping cough globally and in the United States, and also potentially for other pathologies that are not quite recognized yet. Subclinical colonization is pervasive globally. You can go to any country, swab people's noses, and at least in the countries that it's been done so far, it's been pretty consistent. There's a very high rate of background pertussis floating around. Not surprising, because the current vaccines don't block colonization, they don't block transmission, they don't block the reservoir. From the reservoir, you get the escape mutants, you get transmission, and potentially other pathological problems. But BPZE1, because it gives great immunity above the neck and below the neck, is likely to be able to solve the problem of pertussis and whooping cough once and for all. 100 years ago, they started those studies, and with the vaccines for pertussis. And here we are 100 years later with a very significant problem. And it is our hope and expectation that we can do that. And we're continuing to move the program through the various FDA phases. And I hope that you can see why there's good reason for public health to be excited about what is being accomplished with this vaccine and its prospects. We still have a distance to go, but we've made tremendous progress, and the vaccine has performed beautifully. And God willing, we'll get there. So thank you. Thank you for your time.